Good afternoon, everybody. I'm very happy to have with us today in another HIAAD Bridges seminar, Professor Henrietta Nielsen. Henrietta is a great friend, knowing each other since our PhD time, a few years ago. And she's Associate Professor of Neurochemistry and Molecular Neurobiology at Stockholm University. She is also the perfect person, I think, to be here today with us in a HIAAD seminar series called Bridges, because she's working in Europe, but she has been working also many years in Mayo, and she is the bridge between the United States and Europe, because she's doing a lot of different things. I'm going to refer only to a few of them, like she's the senior editor of Molecular Neurodegeneration, a very good journal, as well as she is executive organizer of Alzheimer's disease and Parkinson's disease meeting. That's only two. There, she can tell you more about other things interesting, and that will be helpful for different people, I think. So without saying too many, Henrietta, thank you for accepting to present your very interesting work, both clinical and experimental work in the HIAAD Bridges seminar. And the floor is yours.
Great. Thank you so much for the kind invitation. And I'm really excited to give, you always have smaller talks here and there, but a longer talk like this gives me the opportunity to elaborate on the background of basically how we tuned in on the liver. And I will keep my talk for 40 minutes about. I don't have too many slides so that we can have a discussion afterwards. All right, I will start sharing my slides. Let me see. There we go. All right, so just like Ioannis said, I'm based in Stockholm, so Stockholm University. We have a picture here. This is just very recent. We still had a little bit of snow. We have had very little snow this year around. It's been unsurprisingly warm, but I think that's global. So this is what Stockholm looks like in the summer. So we have winter and summer is kind of melting together soon here. Okay, so today I'm going to talk to you about ApoE. The main topic that my lab is working on. We're working on translational neurodegeneration here at the Biochemistry and Biophysics Department at Stockholm. And we have changed area a little bit, but and I know we've had this discussion in the past, Ioannis, about Alzheimer's, whether you are a Baptist or if you're a Taoist, and we never really took a stand on what we are. So for now, we keep agnostic still. So I want to point out, and for those of you who have seen my talk sometime, I apologize, but I really like to show a few of these slides to position ApoE. So ApoE in humans is quite special because it's polymorphic. So most other animals or mammals, I think actually all of them, according to literature, have a monomorphic ApoE gene, so not polymorphic. Sorry, I'm just going to see if I can get a laser pointer. So here on top, you can see that the common variants in the ApoE gene in human is the E2, E3, and E4. They're quite similar once they're expressed. So the isoforms really just differ at two positions. So there's only at 112 and 158. So they're very similar, and it's very difficult to use antibodies to detect them, the individual isoforms, because of their close similarity. So the ApoE3 variant is the most common one in the population. I'm going to talk a little bit more about that. ApoE4 is not an infrequent, so it's not a rare variant. 15 to 20%, depending on where in the world you are, carry ApoE4, whereas ApoE2 is quite rare. Homozygous individuals are quite rare, so 2%, 1 or 2% of the general population. And also illustrated nicely in this table, I really like it, is that you can see that in other animals, for example, our common lab animals, mice and rats, they have arginine at the two positions. So their ApoE, sequence-wise, is actually quite similar to the human ApoE4. So that's something to keep in mind. And I put in this quite nice illustration as well, because people don't think of ApoE4, for example, as the ancestral variant. So ApoE4 is actually what was the very first variant, and that's also what remains in the animals. For example, non-human primates. It's still ApoE4. sequence-wise, right? But ApoE then evolved into E2 and E3 sometime between when the chimpanzee lineages split from the humans, and the early ApoE4 variant, and also the one that exists in other mammals, is considered to be functionally very similar to the human ApoE3. So sequence ApoE4, but function more like ApoE3. And the evolution of ApoE4 into E2 and E3, combined with also a difference in lifestyle, has been hypothesized to be part of the reason for the prolonged lifespan in humans. And I think also quite interesting to point out that it appears, according to literature, that ApoE4 is more and more, due to selective forces, then pushed towards ApoE3. So it has been the major one, but is then replaced by ApoE3. And I mentioned in the beginning that roughly 15% of the general population carry ApoE4. That's like a truth with a little bit of adjustment depending on where in the world you are. So here we have European populations, for example, and we can see that indigenous people in Sweden, for example, have a very high prevalence of ApoE4, some 30%. Swedes in general have quite a lot of ApoE4, some 20%, or Scandinavians in general. But if you then look at a gradient down south in Europe, we get to Greece, for example. That's just really a fraction of the ApoE4 frequency that we have in northern Europe, which is quite interesting because ApoE4 specifically also is implicated in a lot of different things, depending on sun exposure and whatnot. And then there's also a few other, this is a very nice publication, quite old, but I think the numbers are still quite relevant. For example, other countries, Japanese, 10%, but you can also see that there is quite some variation in the African populations with up to 40% in pygmies, for example. So I think this is quite interesting to keep in mind when we do clinical studies. Where do we have the ApoE4 when we do our clinical studies? Most of you, if it's an Alzheimer's community, know that ApoE4 is the main genetic risk factor for sporadic, what we still call sporadic Alzheimer's disease. But it's also the main genetic risk factor for a closely related disorder, dementia with Lewy bodies, which I find quite interesting and that I tend to call as a disease in the twilight zone between Parkinson's and Alzheimer's disease. Interestingly, so the ApoE2, again, so the most rare variant seems to be protective, similarly protective against both these disorders, so both dementia with Lewy bodies and Alzheimer's disease. Whereas homozygous individuals have more or less 15-fold higher risk of developing Alzheimer's disease and roughly 6 times higher risk of developing dementia with Lewy bodies. And coming back to also the different areas in the world, it's quite interesting to see that the actual risk imposed by ApoE4 for Alzheimer's disease varies across populations. So Africans, for example, this is also a study that is about 10 years old or so, and there is more and more studies showing relevance of ethnicity and race in the ApoE4 promoted risk of Alzheimer's disease. So we can compare, for example, in this study, Nigerians that had a 1.68 relative risk in E44, whereas, as I mentioned before, 12, 13, 15-fold higher risk in, for example, Scandinavians, US Japanese folks, as in this study, 33-fold higher risk. So there is a huge difference in the risk imposed by ApoE4 for Alzheimer's disease. There's been quite some studies coming out too, talking about African Americans, for example, and also US Hispanics. And here in this table, there is some 2 to 6-fold higher risk in E4A4 carriers, but some studies even suggest that there is no risk or no higher risk of of Alzheimer's disease in ApoE4 carriers in some of these populations. So ApoE is expressed in many different places in the body. We normally tend to think of it mostly in the brain, but it's also expressed in endocrine tissues. We also have a lot of expression in the liver and the gallbladder. And we also need to think about other cells like monocytes, macrophages, which are, for example, very important for other diseases like atherosclerosis. The bulk of ApoE that's circulating is produced by hepatocytes in the liver. The bulk of the ApoE and the central nervous system, we tend to think is produced mainly by astrocytes, but also parasites produce quite a bit of ApoE. Microglia produce ApoE. And what's important to take home here is that there are several liver transplantation studies showing that the peripheral pool of ApoE does not match with the central nervous system ApoE pool. So it's not supposed to, or it does not necessarily cross the blood-brain barrier. I got to move you guys a little bit so I can see my screen. Let's see. So the protein is some 299 amino acids, roughly 35 kilodaltons. And as also illustrated by the name, it's a very important lipid carrier. However, the different isoforms are equally well equipped for this. So ApoE4 is considered to be a poor lipid carrier. And I mentioned that there are two different pools of ApoE in the central nervous system and in the periphery, and they are also slightly different. They're differently sialylated. We can see here, so CSF is more sialylated, whereas in plasma, there's less sialylation. It gets cleaved off once it's secreted. And there's another difference. So whereas plasma ApoE can be mainly found in HDL and VLDL, ApoE4 predominantly in VLDL and LDL, the central nervous system ApoE is mainly found in the bigger or the HDL particles. There's also a study showing that the plasma ApoE levels between different isoforms have a different turnover rate, that ApoE4 has a higher turnover rate than the other isoforms. However, that was not shown to be the case in central nervous or CSF ApoE levels. Here's just also some illustration of the two main domains, so the receptor binding domain and the lipid binding domain. And we also have a few mutations that have been mentioned in the literature. Christchurch mutation, maybe most of you have heard about, but there is also a Jacksonville mutation discovered a few years ago. So how does ApoE and ApoE4 relate to many of the different features of Alzheimer's disease pathophysiology? I'm sure most of you have seen this figure. It's also still very relevant. So just considering the different aspects hereof, we can summarize from the literature that ApoE4 is predominantly related to amyloid beta deposition. So regardless if you have Alzheimer's disease or not, or any cognitive, I should say, symptoms, ApoE4 promotes the deposition of amyloid beta in the brain, and also at an earlier age. It's also been shown that ApoE4 carriers exhibit brain insulin resistance, and also brain, so glucose metabolism is altered in some way, and that's even in healthy individuals. There's also been imaging studies showing that ApoE4 carriers have structural and functional brain differences of various ages. And I mentioned the Christchurch ApoE3 mutation appears to be very protective against the development or the course of autosomal dominant Alzheimer's disease, which normally occurs quite early in life. But in this one case that was described, the lady actually did not develop any dementia symptoms until her 70s, which is incredibly rare. So it also illustrates how important ApoE is to Alzheimer's disease per se. It's a key player. The relation to tau pathology is becoming more well known now, but it's still not clear. There's been suggestion that the lower heparin affinity, as in Christchurch mutation, for example, seems to be beneficial. So ApoE and ApoE4 is not only relevant to Alzheimer's disease, it's also relevant to other diseases. And this is from a review that we published a few years ago. You may have heard it's of interest for the blood-brain barrier function and integrity. It's also has some importance for stroke. We have MS and ALS, microglia activation, which we also know happens, of course, in Alzheimer's disease, which is the main feature here. I'm going to mention ALS a little bit more later on when it comes to plasma levels of ApoE, for example. But this is really just to illustrate that we need to think of ApoE4 with numerous functions of relevance to not only Alzheimer's disease. All right, so in my team, we are quite interested in the peripheral ApoE component, and which is maybe strange because I just mentioned that it does not cross the blood-brain barrier. But many, many years ago, when I first started study Alzheimer's disease in the relation to ApoE in more detail, I was interested in biomarkers a lot. I still have an interest in looking at what the situation is like in humans, the plasma levels versus the cerebrospinal fluid levels. And we published many years ago, I will get to that, a quite striking feature of ApoE4 carriers in plasma. But I want to show you this paper. There's a few papers coming out that came out of Denmark, where they actually have assessed many thousands of people. And here in this particular graph shown that the individuals that have the, lowest levels of ApoE actually have the highest risk of developing not only Alzheimer's disease, but also all types of dementia. And the authors, so I'm paraphrasing here in this context, stated that only a 1 milligram per deciliter decrease in genetically determined ApoE levels increase the risk of dementia 2 to 4-fold. And the 2 to 4-fold is really on par with carrying one ApoE4 allele in terms of risk of Alzheimer's disease. And then there's been numerous studies showing, including our own, that plasma ApoE levels appear to be modified by ApoE genotype, possibly also sex, but also disease status, which is not 100% concluded yet, I'd say. And also it depends on what population you're studying. So the first part here, we can see here the E4E4 carriers have the lowest levels of plasma ApoE. We have also shown in several publications that ApoE4 carriers, and this is actually not a huge difference, but there is a significant difference between E4 homozygous individuals and the ones not carrying ApoE4 at all. We've seen that there might be some difference in between diagnostic groups. MCI, Alzheimer's disease, dementia, however, that is mainly driven by the presence of the ApoE4. Interesting though, but also again, the ApoE4 carriers are in this group. Those with amyloid beta positivity or amyloid beta pathology in the brain also appear to have the lowest levels of plasma ApoE. So, and I mentioned that the risk of Alzheimer's disease in ApoE4 carriers appears to be different between ethnicities or people with different race. And we've studied a few years ago together with a scientist at Rutgers University, a cohort of diverse patients, so both Black African Americans and non-Hispanic whites. And here we could find no difference in between diagnostic groups, whereas we in the non-Hispanic whites could see a feature of reduced levels of ApoE and the ApoE4 homozygous individuals. Whereas although we have a very small number of E4 homozygous individuals in the Black African Americans, it seems like at least there is no impact. If anything, it seems to be higher levels of ApoE in that particular group. So this is results that we still would like to follow up more closely. And I mentioned also that ApoE is mainly derived from the liver in the periphery. And otherwise, many people ask, why would you be interested in the liver? We have the blood-brain barrier. Most things are shielded off. But just taking a quick look at PubMed even today, there is more and more studies popping up that actually has the liver even in the title. So we have, for example, just coming out 2025, liver function and Alzheimer's brain pathologies. We have liver as a new target organ in Alzheimer's disease. People do talk about amyloid beta degradation, for example, which is a very important feature of the liver. There's been also a study showing that the amyloid beta degradation is in fact impaired in Alzheimer's disease, and also the amyloid beta degrading enzyme levels were disrupted in livers of Alzheimer's disease patients. There's been an interesting study showing that liver-restricted production of the human amyloid beta could actually promote accumulation of human amyloid beta, both in the vasculature and in the brain tissue of the specific mouse strain. There's been several studies showing a correlation, at least, between plasma levels of liver enzymes, so liver function, to Alzheimer's disease biomarkers, but also to cognition. And there's also, in my view, a quite interesting feature of midlife non-alcoholic fatty liver disease and the increased risk of dementia. There's been several studies also pointing out that the amyloid beta degrading capacity of the liver might be distorted once you have a fatty liver. So I think together this really paints a nice picture of an organ that has been understudied, in my view, for a long time. So we have studied the liver for quite a while. We're not hepatologists, we're not liver experts. We have very good collaborators with people with access to donated liver tissues at the Karolinska Institute here in Stockholm. And this is already several years ago. We did a gene expression analysis of biopsies, liver biopsies from ApoE4 carriers and non-carriers, and found that quite a lot of genes were differentially expressed between these different genotypes. And we used the extremes, so the E2 protective and an E3 considered to be neutral when it comes to risk of Alzheimer's, and a neutral, and then the risk allele ApoE4. And at least on the ApoE mRNA level, it seems like there was no difference in the expression of ApoE. We still to this day have not been able to produce any allele-specific expression levels. So if someone is working on that, that would be very interesting to hear what you guys have found. So looking at the same, so basically not the same. So these were biopsies, but in hepatocytes donated from the same source, I'd say. So looking at the protein levels, we could, however, see that the ApoE3, E4 hepatocytes had lower levels of the protein ApoE. So expression and the protein levels don't necessarily go hand in hand. We've also done some network analysis and found that, as you can see, we have our ApoE here, but there's also numerous different players involved. So ApoE expression per se may not be the culprit or the main feature here that we need to focus on, but also downstream effectors should be considered. So again, we are trying to characterize the liver with quite agnostic view in terms of really just ApoE4, non-E4, to see if we can find any signatures that would be of relevance to study further in animal models, for example, or in vitro cultures. So one of our recent publications is on the hepatic lipidome and signatures of hepatic ApoE genotype. And here we also looked at primary human hepatocytes, so hepatocytes isolated from donor tissue from the Karolinska Institute. So these are never cultured hepatocytes. So the profiles are not affected by culture conditions, culture media, and so on. They were directly isolated from the liver tissues and then snap frozen in pellets. So they're hepatocyte pellets. And also here we looked at the extremes, so E2, E3, and E3, E4, but we also looked at the E3, E3, and E3, E4. And found that there were quite some interesting findings in terms of mainly acylcarnitine, but also acyl-PG, and that seemed to be increasing with the presence of E4 or the lack of E2. Not sure which. Some of these were related to mitochondrial membranes and so on. So mitochondrial biology. And we're trying to figure out more further how to interpret this data. So if anyone is out there and an expert on lipids, it would be interesting to hear from you. Our next step in the characterization of the liver is to proteomics. So more or less the same hepatic pellets that we have used for our lipidomics, we have also done proteomics on. And so by now we have accumulated some 75 donors. And out of these, they actually follow the distribution of the ApoE genotypes in the population quite well. So these are individuals that come to the hospital to either have a liver transplant, so their livers are explanted, they get a new one, or they're liver donors, whereof the livers might not be matching with anything, or there's other issues, and then they are used for the isolation of these hepatocytes. But we've collected tissues and hepatocytes ever since 2016. So far, we've only had ever one E2E2. We've had several E4E4 actually, but for this study, we only had cells left from one donor. But you can see that the E3E3s are the most common ones, and then the E3E4 is the second most common genotype in our material. So 24 out of the 75 were ApoE4 carriers. Similar distribution between the sexes, so females and males. Average or median age 59, and we had a very huge range of the age, all the way from 1-year-old to 86-year-olds. So this is really just a very preliminary volcano plot. This is unpublished data. My student Maja Sjödahl will be presenting in more detail the more preliminary data at the ADPD conference that's coming up in April. So if you're attending, you should go see her poster. But we can see already that there seems to be quite some proteins identified, so that seems to differ between these hepatocytes with and without an ApoE4 allele. Maybe you have a favorite molecule. We haven't found one yet, but we are continuing our analyses on this. So another line of our studies is to actually, as I mentioned, trying to model this in vivo, and that's quite tricky. The mouse liver is quite different from the human liver. The mouse lipid metabolism is very different from the human metabolism. And so we have actually by chance stumbled onto this quite exquisite animal model, which is a humanized liver mouse model. So it's the FRGN humanized liver mouse model, which gets transplanted with primary human hepatocytes, and which will then repopulate the human mouse, the mouse liver. It's also, so I forgot to mention, it's a triple knockout mouse, which is highly immunosuppressed, and it also has a Fa deficiency. So when you don't treat the mouse with a drug that it needs for its liver to work, then the mouse hepatocytes will die. They will die from hepatotoxicity. And by injecting the mouse with primary human hepatocytes into the spleen, they will then find their way to the liver, repopulate the mouse liver that is dying, and then basically transform the mouse liver into a human liver. So it's a quite cool system, quite expensive system, but quite cool. And this is also a proof of concept model to us to study liver-derived ApoE in a species-specific physiological environment, because it's not just ApoE, but it's everything in the liver around ApoE. So this mouse model will have a human lipid profile in the plasma, for example. The ApoE will be expressed under the human promoter, under the human regulatory mechanisms. So this is a study that we published a few years ago, showing that the plasma levels in these mice, and again, we have very few N numbers here because it's quite a big undertaking to first of all, have primary human hepatocytes, which is needed to transplant these mice and to have them survive. And then also the cost of them are quite expensive. Here we had 1, 2, 3, 4 E2, E3s, and then we had E4, E4s. And we see that the plasma levels of ApoEs here are quite similar to those that we can find in humans. And since they really just express the ApoE in the periphery, they do have their endogenous mouse or rodent ApoE in the brain. And we were quite interested to see what the levels of those looked like. And they were quite similar. There was a slight difference in some of the areas, mainly hippocampus, between the E4 carrying mice and the non-carrying mice. But mainly, I think this is interesting and probably goes quite well with other studies as well, that the hippocampus has very high concentrations of ApoE in general. So we looked at a few different parameters in the brains of these mice that had the human ApoE4 liver or the E2, E3 liver, and found that there were a few differences. And we've illustrated some of them here where we had increased and decreased or near significant differences. We were interested in mainly synaptic proteins, for example, but we also found that there were some differences in terms of the astrocytic marker, GFAP, microglia marker, and also depending on what brain areas, so cortex, hippocampus, thalamus, for example, or some other brain areas we looked at. And then trying to associate the plasma levels of ApoE4 specifically, because we had 7 of these animals, it appeared like higher levels of ApoE4 in general were correlated to negative outcomes. So lower levels of AMPA receptors, of these synaptic markers. So just because there are or there might be lower levels of ApoE4 in the periphery, that does not necessarily mean that those levels need to be pumped up, because that might also not be good. So it's quite a difficult nut to crack. We've also followed up on ApoE3 humanized liver mice specifically, and ApoE targeted replacement mice. So these are the conventional ApoE mice. And this is a study that we published from last year. I was about to say last year, but it's 2025 now. And we can see that the liver weight, which was not completely unexpected, was bigger. So these humanized livers were quite bigger than the ones we could find in the targeted replacement mice. But in the targeted replacement mice, you also had slightly bigger in males, which might not be that difficult to comprehend either. Plasma levels, however, were not different between the E3 targeted replacement mice, females and males, whereas there seems to be a huge difference in the female males. And we're not really sure exactly why this is or if this is true, so we are following up on these results as well. We were also very interested, I'm sorry this looks a little scattered, to correlate the plasma ApoE3 levels to behavior outcomes and also to cognition, memory outcomes. And here is just an illustration. I'm not going to linger much on the results, but the plasma ApoE levels, again, these are E3 plasma levels, seem to be significantly correlated to a few different outcomes in these in a big behavior and memory test battery that we used together with Jacob Braber at OHSU in the States. Basically, a few of them seem to be either sex-specific, but also model-specific. So some of them were present in the targeted replacement mice, whereas they were not present in the humanized liver mice. So trying to wrap up a little bit. So we have currently, I'm backing up, also tissues, livers, and plasma from ApoE4, E4 humanized liver mice. We transplanted a total of, I think they were 28 animals. On the 5th of July, 2023, they were sacrificed last year. And out of all of the ones remaining, I think we have 5 or 7 left. It's a very tricky model to work with. It's very difficult to foresee which animals will properly become humanized, because you need to have the liver humanized in order for the mice to live because of their FOD deficiency. So it's a very tricky model to work with. I'm not sure it's a good model altogether, but this is what we have for now. All right, so I know that there is a few clinicians in the audience as well, and I also wanted to bring this up a little bit. So ApoE genotype and clinical relevance. Well, so we tend to think of ApoE for Alzheimer's specifically, but there might be relevance to also other diseases. And I think most of us know of the cardiovascular disease association with ApoE4. ApoE4 individuals tend to have higher plasma LDL, so bad cholesterol, and also downregulated LDL receptors. So as an ApoE4 carrier, which I am, ApoE3E4 is my genotype, I need to have my cholesterol levels checked every now and then to make sure I'm on track. Several studies have shown that ApoE4 carriers also exhibit lower plasma levels of the acute phase reactant C-reactive protein. I think this is very interesting because ApoE is also a quite important immune modulator, and that was quite evident also during the COVID pandemic, where there's studies showing that both the risk of severe COVID and also the severity of COVID seems to be higher in ApoE4 carriers. There's also numerous studies showing in general ApoE4 susceptibility to viral infection, and also ApoE4 enhances HIV infection and progression. However, when it comes to the liver, it seems like ApoE4 is protective against hepatitis C and B mediated liver damage. What I think that most of us now during the last couple of years think of when it comes to ApoE4 and clinical relevance is really the risk factor of developing adverse reactions like the amyloid-related imaging abnormalities, or ARIA, due to the anti-amyloid beta immunotherapy. And this is a table compiled by from the literature. So really just showing that the frequency of ARIA-E, so the less aggressive side effects, I'd say, is very common in ApoE4 homozygous individuals. So a third, and sometimes even up to half of them develop ARIA-E, even if it's transient. But ApoE4 carriers, who are also the ones that develop amyloid beta pathology at an earlier age, and that in my view might be the ones that really need the therapy even at an earlier age, are the ones that also have the highest risk of side effects. So I also wanted to illustrate that we don't really know if plasma ApoE levels play a role into this risk. So to summarize, ApoE4 may produce alterations in plasma ApoE levels, and these may then be relevant to the risk of neurodegenerative diseases, and that this risk and potentially also the plasma levels seem to be somehow ancestry, race, or ethnicity dependent. We use humanized liver mice to study ApoE4-driven phenotypes and consequences to the brain. So a proof of concept where you have human ApoE produced under physiological mechanisms, human mechanisms in the periphery, but not in the brain. We have several findings pointing to that the plasma liver-derived ApoE links to various processes in the brain without actually crossing the blood-brain barrier. And our ongoing studies appear to support that there are some ApoE4 phenotypical traits of clinical relevance, mainly in the liver. And as I just mentioned to you, that ApoE4 is associated with an increased occurrence of adverse effects to the anti-amyloid beta immunotherapies, but the molecular underpinnings we don't really understand yet. But together, this would suggest that modification of plasma ApoE levels and potentially also the association with lipoparticles may be of interest to clinical parameters, including those to eliminate or reduce the risk of Alzheimer's disease. So I would like to acknowledge this is just some of our collaborators, some of the funding. I've highlighted two very important individuals. They have already left the lab, but they have produced most of the results that I've been showing now. Andreas Ioannis, Kalisharan Patra, who both have moved on to other positions. I think Andreas is back in Greece by now. And then I'd like to take 2 minutes, maybe 1 minute, to also promote a other webinar seminar series that's from the International Society for Molecular Neurodegeneration, or IceMind. Since a few years now, we started IceMind in 2021. I'm leading the organization. I'm the current president. And every month, we also broadcast science webinars, and the most not recent, the one to come up soon is February 20 with Inemuk Young from Korea. And then we have 2 European speakers. We have a lot of people from all over the world speaking, trying to accommodate the time difference. It's free of charge to participate, but you need to register. And then you can also watch the recorded webinars online if you are a member, and you can become an IceMind member on our website. It's free to students, so I would definitely encourage all students out there to become members. And I think this is my last slide. I want to also promote the upcoming IceMind Next Gen 2025 edition. It will take place online on the 25th and 26th of August with an abstract submission deadline in June. Registration and abstract submission is free for IceMind members, and we take $50 for non-members, so not a huge cost. And it's mainly to broadcast science of young scientists in the field of neurodegeneration. And we had our first one in 2023, so this is our second IceMind Next Gen 2025, and I'd like to invite you all to participate. Great, I think I'm done.
Thanks
Thank
a lot,
you.
Henrietta. It was a great talk, easy to follow as usual, and very entertaining, and we learn a lot. So are there questions from the people who are in the Zoom? And let's see if there's any questions on YouTube. So any questions? Otherwise, I have plenty, but I prefer to give the, okay, Angelica, for sure. Go.
Okay, it's a great talk.
Stop sharing, Henrietta. Can you stop sharing?
Oh, sorry. Yes.
Thanks a lot.
There we go.
Great talk. Thank you very much for this very nice presentation. I enjoyed it very much. So I would like to ask, I may have missed it. Did you check whether ApoE can be found? I mean, the humanized, in your humanized model, did you check whether ApoE is detected in the brain? Because I'm asking because, let's say, ApoA1, it is proposed that it can cross the blood BBB. So, and so did you check in your experiment?
We did check. Yes, we did check, and we did not find any. But I cannot guarantee, at the level of detection level, that nothing is there. But we didn't find any.
And also there is this study 2022 that they had this model that was expressing ApoE3 and ApoE4 in the liver, but not in the brain. And again, they also found that peripheral ApoE affects Alzheimer's disease pathogenesis. And I think they were able to detect ApoE in the brain, but I'm not sure.
I think the vasculature, if that's the same study that I'm thinking about, Gujanbu study.
Yes.
Yes, exactly. So I think they mainly found vasculature impediment. So we have not looked at vasculature. really, since we have the tissues now, we can look at it. And I think what's really important too is to have the whole lipoparticle composition, because even if you express the human ApoE, only the human ApoE gene in the liver, you still don't have all the lipoparticles that we, like the HDL, LDL ratio, for example. So that's what we're trying to overcome, even though it's a difficult model with mice not having LDL, for example.
Yeah, that's a problem also.
Yeah.
Maybe you could use, let's say, a CETP mouse model or a Leiden or something like this.
Right. Yeah. So this particular mouse model has been used quite a bit to study liver diseases, and it's mainly, so we've been working with Yacuris in Oregon for many years now, a company that has developed these for us. And at least in their information to us, the people normally using them are pharma companies. Basically, because of the cost, they're extremely expensive. I think they're $6,000 per mouse, so they're very expensive. And then you have to keep them, and they're very sensitive, immune suppressed. So I don't think it's a good model long term. It's very tricky to work with them. But they've been used basically for toxicology studies and so on, but not really for any brain related studies.
I see. Okay, great. Thank you.
Good
luck with the rest of it.
Thank you. Thanks.
Is any other questions? I can ask, because I know that other people are also listening from the YouTube. But Henrietta, do you really think? I know there's a lot of amyloid beta, and I saw recently coming from Tau some good publications from Hotzman Lab and others, really good names and really good work. So how do you consider, the impact on amyloid beta is okay, it's there. However, if we consider that Tau is the final executor in the neuronal degeneration and we need Tau for the killing of the cells, would you see, first of all, what is the molecular connection on that? If you can share with that, that's the first part of the connection. And would you see an ApoE positive role, detrimental role or risk factor role in other tauopathies? Is any relationship to that direction?
Good question. So to your first part of the question, so I do think that the link might be amyloid beta. That's one thing. So as the initiator, and then Tau comes in next.
Okay.
So there is several. So we're working on a different line thing and work in the lab too that I haven't mentioned. It's hepatic amyloid beta degradation, and there is actually a few studies coming out now with amyloid beta. Once it's cleared from the blood over the blood-brain barrier, 60-70% is cleared by the liver, and it's quite a fast process. So if you do have a malfunctioning liver that's struggling with lipids, for example, and other things, then this degradation might be hampered, and you get the zinc hypothesis is quite old. People have been talking about this for a long time. You're trying to clear things. I'm not sure it's that simple. It's the bidirectional communication across the blood-brain barrier, where you also might have peripheral ApoE sitting in the vessels and also there. So not just possibly hampering the amyloid beta degradation that does come across in the liver, but also potentially clogging some of the vasculature to enable the clearance of amyloid beta across.
You point out, I think, the most interesting part of your work, which has to do that the ApoE4 in the liver affects without crossing the brain. And the work of Andreas Genedis in this molecular psychiatry, I think that's why I see a lot of recognition of the work, because of that novel thing you prove with this work. And I see Andreas is in the audience.
Yes.
So Andreas is in
awesome.
The audience. It's the only PhD student you had, or you have another one, I don't know, but at least you had one. That's good. So back to the question. This is really amazing. Without crossing to the brain, it does so many changes. So do you think there is that the liver sends some messages through the blood, through exosomes, that we're working more and more people here also? What do you think would be the possible communication between the liver under the ApoE driven malfunction that you saw is very nicely the effects on the brain? Because it doesn't cross. You saw it very clear. It doesn't cross. The ApoE itself from the liver doesn't cross. So what is the mechanism? Any ideas, any suggestions, something you're working that you can share with us for sure?
So not to this point. Oxysterols have been, there's a lot of this communication back and forth. And when you talk to hepatologists, everyone knows that if you have a malfunction in liver, people that come in for liver transplantation, they almost all have encephalitis. There is this bidirectional, and so this is not strange to people that work with liver diseases. It's strange to us that work with a brain disease, right?
Exactly.
So I'm sure clinicians in the audience might be able to explain more. But I do think that this whole homeostatic process, once it's tilted in the periphery, and I do think that the liver lipid metabolism is deeply implicated in this, not performing well, and then that sends signal back to the brain. But we're trying in every way to target that. We're going from large, like doing these characterizations of lipidomics, proteomics, and then hopefully doing multi-omics studies to pinpoint where exactly we're going to put our money to figure that out, more or less.
The only way to communicate between brain and liver, it is the blood, and the BBB. So something has to go through this.
Exactly. So we have done studies on not a very sophisticated blood-brain barrier model. We've used primary human microvascular endothelial cells.
Mm-hmm.
And we had a student a few years ago. This is unpublished data, so this is something we want to follow up on too, is that the blood-brain barrier, or at least endothelium cells, do take up ApoE, at least in culture. But it seems like they take up less ApoE4 than ApoE3. And once they have taken up ApoE, it seems like, at least for ApoE3, that it's beneficial for the integrity markers of claudin, occludin, and so on. So, a speculation would be that ApoE4 is for some reason not properly there or not taken up, and therefore the blood-brain barrier integrity might also not be as well performing as has been published many times before in E4 carriers. Not necessarily disrupted, which will give a lot of problems, but just not to the level of where it should be.
Okay, that's a nice approach. But anyhow, I see Vaso.
Yeah.
Because that's completely changed the way we see brain malfunction and brain aging through the liver malfunction that has through metabolic or signal interaction. That was really like what I know your work, but more and more I see the coherence where you develop and there's a lot of clinical and preclinical data suggesting and supporting that notion or that hypothesis. It's really interesting for me. I also don't know anything about liver, but I don't know if Kostas or any clinician has any other suggestion, or Ioannis want to have.
If you completed this set of discussion, Henrietta, thank you very much for your very informative presentation. I'm not an expert on ApoE or ApoE4 specifically, but I have done a search for any potential correlation between the expression of ApoE4 and major neurotrophic factors like BDNF. And there are very few papers in the literature, many years ago, actually, before 2015. So do you know, or did you find any correlation between even the liver ApoE4 and the changes in neurotrophin or neurotrophin receptors expression in the brain?
We haven't looked yet, but I'll add it to my list for sure. So the very first study that was published with Andreas' work, when we got the brains, we were trying to really just probe different areas this time. So when we actually have transplanted mice, we have all the tissues, we have plasma, we have the livers, we have behavior, we have memory, cognition. So this time we know where we're gonna search, and I think that's a good target to look at too.
just mention here a very recent finding that we have, which we haven't go in depth to search for it. You know, in human iPS cell lines and derived neurons that carry the ApoE4, and only at the ApoE4 case will fully lose the expression of a major neurotrophin receptor, while the receptor is present in PSEN 1 or 2, you know, mutations in human, in ApoE4 is totally disappeared.
Interesting.
We haven't done anything yet, but
Okay.
you are the person perhaps that will be some kind of useful, this kind of information.
Interesting. Thanks for sharing.
And
We can discuss it sometime.
Talking about iPS cells, because that's what we are doing.
Introduction, Vasa, it was a nice introduction.
Exactly. No, I haven't checked, to tell you the truth, what's going on with ApoE4 in the liver. And but I was wondering if you're familiar with any iPS stem cell derived studies related to ApoE4 liver, because listening your excellent talk, I was thinking maybe some co-cultures of neurons and the hepatocytes, and be able to see how this transfer and what are the interactions and how we can check the mechanism there. But I haven't checked for the liver, to tell you the truth.
I think there's very little out there. I know it's fully possible to make hepatocyte spheres, for example. People do these organoids and so on. This is something we would like to explore as well. And that's fully possible with iPSC cells as well. I can't tell you any direct studies that come to my mind. It's quite sparse. And I'm really surprised because there's a lot of discussion with the lipid metabolism, atherosclerosis, and where you have all the lipids and ApoE in the periphery. But surprisingly few studies focus on liver.
Yeah, I think because, everyone is focusing, including us, of course, on neurons, astrocytes, brain organoids, different types of brain organoids, and also brain-gut axis. But this, it's a brain-liver axis, so it's something new relatively. So I guess in the coming years we will see more and more publications.
I think so. I mean, I can see the field is really more and more. I'm happy to see that the number of publications on liver function, even, and both clinical and experimental studies is going up for Alzheimer's disease. Yeah.
Thank you. Thank you very much.
Thank you.
Indeed, Professor Kostas Liketos from Johns Hopkins also, he had to go because he had to see patients. He said that there's a lot of possibilities on clinical side too. And anyhow, it was, we could discuss here for ages, and I know that this is a really hot new topic opening to most of the brain people. Probably for the liver people is much more familiar, but that's why we're here. And I think that will be a nice forum for interactions, and everybody who likes to work or interact with Henrietta. Stockholm is not so far, and she was here for the ISMN meeting like 2 years ago when me and Christina were happy to help organize the local organizing committee here.
Hey, Christina.
Soon she will be back.
I didn't have the time to.
And we will have to invite her to give a talk in the next Genedis meeting. I'm sure she will be a great, great speaker and great work. So thanks again, Henrietta, for being with us. And for all of us, we will meet again in 1 year, as usual, the first Thursday of the month with another HIAAD Bridges seminar. Thanks a lot, and have a nice afternoon.
Thank you.
Thank you.
Thank you.
Thank you.
Thank you.
Henrietta, really great talk. Now the link is off.
Okay, great.